All right, so we've reviewed glycolysis, the oxidation of pyruvate, and the citric acid cycle. All of those processes produce a product or products that will eventually be utilized in oxidative phosphorylation. Now remember that the citric acid cycle and glycolysis phosphorylated ADP to ATP by means of substrate level phosphorylation. Here we're going to perform a process called oxidative phosphorylation. And the thing you need to remember about this process is it's broken into two separate components that are going to work in tandem. The first component of this process is called the electron transport chain. If we look at membrane, in the mitochondria, what we will see is we will see a series of proteins that are going to be responsible for transferring electrons from protein structure to protein structure. Okay, so we've got protein complexes responsible for transferring electrons. And what you need to know about the way this chain flows is that the protein complexes and the members of those complexes that exist at the beginning of the chain have a lower electronegativity than those towards the end of the chain. Okay? So what will happen is all of those electron carrier molecules that we produce, the NADH and the FADH2, okay, that we produced during glycolysis, the oxidation of pyruvate, and the citric acid cycle, those are going to come and donate their electrons to the chain. So they're going to be oxidized. So the electrons are going to transfer from the NADH or later, the FADH2, thereby oxidizing those molecules and transferring their electrons. Their electrons will be transferred through the chain, and ultimately, because we're talking about aerobic respiration, those electrons will eventually, at the end of the chain, be accepted by oxygen. Oxygen will have the highest electronegativity of all of the components in this chain. Oxygen will be the final electron acceptor, and it will combine with protons to form water. Okay, so oxygen is the final electron acceptor. It combines with protons and forms water. So there's our water that was produced during respiration. Now, the interesting thing is, as these electrons move through the electron transport chain, as they move through these protein complexes, they result in the pumping of protons across the inner mitochondrial membrane. Now, you'll recall the electrons have a negative charge. As they move through these protein complexes, they give the protein complexes a negative charge and attract protons, which results in the pumping of protons across the mitochondrial membrane. What this does is it generates a gradient of protons. So we have a really high concentration of protons on one side of the membrane compared to the other. And you'll recall from the diffusion unit that when we have a really high concentration gradient, these protons will ultimately, if they have the capacity to do so, diffuse back across the membrane from an area of higher concentration to an area of lower concentration. Now, because these are charged, they currently have difficulty doing that. Remember, biological membranes are selectively permeable, and the characteristics selected for are the ion or molecule must be small, and it has to be nonpolar for it to move through the membrane. Now, obviously these are ions, so they are charged. They're small, but they're charged. So we need a protein to perform facilitated diffusion. And the protein used in this particular process is used in conjunction with these protons in a process that's the second part of oxidative phosphorylation called Kimmy Osmosis. We're going to use a proton gradient to perform work as these protons diffuse down their gradient. And the protein involved in that process looks like this. Okay? And this protein provides a channel by which the protons may first move into the protein, and ultimately they'll be released via the protein on the opposite side of the membrane. But before they do that, they wind up in these cavities inside the membrane, which is nonpolar, and they cause this protein to move. It spins. And as it spins, it causes another structure in the protein to spin, and that spinning, that kinetic energy, is sufficient to convert a molecule of ADP plus an inorganic phosphate to a molecule of ATP. Okay? So, again, the protons move into this protein, causing it to spin. That proton motive force causes another portion of the protein to spin, and that is sufficient amount of energy to then take adenosine diphosphate and an inorganic phosphate and ultimately convert it into adenosine triphosphate, or ATP. Okay? This molecule, this enzyme responsible for synthesizing ATP through Kimmy osmosis, utilizing this proton gradient, is called ATP synthase. ATP synthase is the final piece to this whole process. We first generate a proton gradient using the electron transport chain and the transfer of those electrons to pump protons across the mitochondrial membrane. And then those protons begin to diffuse down their concentration gradient, but they first must enter ATP synthase. Once inside, they cause it to spin, which is a proton motive force. That proton motive force causes this part of ATP synthase to also spin, and that movement is sufficient to generate molecules of ATP. Now, as we saw in glycolysis, substrate level phosphorylation generated 2 molecules net of ATP. The citric acid cycle, substrate level phosphorylation, again, generated 2 molecules of ATP. Oxidative phosphorylation using the electron transport chain and Kimmy osmosis is capable of generating between 26 to 28 molecules of ATP. So while it was nice that glycolysis and the citric acid cycle were capable of generating ATP, those processes really contributed to this process by producing those electron carriers, which would ultimately donate their electrons to the electron transport chain, thereby allowing us to generate the proton gradient that would be needed to fuel this process here, where we could then use the movement of those protons to perform work that would ultimately allow us to generate adenosine triphosphate. So this is why this process is so important, and why the production of these electron carriers that will be oxidized, thereby donating their electrons to the electron transport chain, are so crucial in the overall progression of oxidative phosphorylation. So that is aerobic cellular respiration.